It's my pleasure to welcome everybody here. I see 54 attendees to the Aquatic Symbiosis Genomics session of BG24. The session is going to be hosted by John Archibald from Dalhousie, Canada, and myself. And we want to talk symbiosis for the next hour. The ASG is known as a flagship project, both of the Sanger Wellcome Institute and the Gordon and Betty Moore Foundation. And together they set the goal to sequence nothing less but 500 symbiosis genomes of hosts and their microbial and eukaryotic partners. In the next hour, you will have 4 speakers covering topics such as methodological development, symbiosis relationships, and implications for conservation and biotechnology. Together as a team, we believe that what we do has the potential to be paradigm shifting in the world of aquatic, and that is marine and freshwater symbiosis. So please join the ride and enjoy it, and feel free to ask questions both by raising your hand and also by using the chat as we go into the meeting. I'm handing over to John, my colleague. Maybe, John, a few words from you before we get started.
Yeah, sure. Happy to just say a few things. Uta, that was a very nice summary of the spirit of ASGP, and the challenge and opportunities that come along with focusing on symbiotic consortia, sequencing them, and trying to produce high quality genome assemblies is immense. I think it's safe to say, and so I'm really looking forward to getting us some updates from the 4 speakers on this topic. I guess we have an hour session in total. That's right, Uta and Hayu. And so we can make sure that the speakers roughly keep to time as we move along. So I won't take any more time. I'll just pass it back to Hayu. Are you coordinating the first speaker slides up?
Yeah, so maybe the first speaker is Graham Oatley from the Sanger Institute. So maybe, Graham, if you could have your slides, and I'll be watching the time. So you have 12 minutes plus 3 minutes discussion and anything that comes on via the chat. So stage is yours, Graham.
Great. Thank you, Uta. Hi, can I share my screen, please? Thank you. Good day, everyone. My name is Graham Oatley. I'm a senior research assistant in the Tree of Life core lab. And today I'll be talking to you about coral and jellyfish wet lab workflows. This is work that has been led by my colleagues Amy and Ben in the Tree of Life core lab, and a lot of the other work that has been done in the core lab on the ASG project is done by many other members of our team. So Uta's just given a nice summary of the ASG project, but in brief, our aim is to produce high-quality reference genomes from a diverse group of both freshwater and marine organisms. And these groups include the algae, the ciliates, jellyfish, mollusks, and the sponges. The samples that we've worked on in the Tree of Life core lab have been collected by 10 global hubs, shipped to Sanger, and then handed over to us in the core lab where we do all the wet lab work. So far, we have worked on 460 symbiotic systems, of which 151 genomes have been published up to date. And as we see every week, there's a couple more coming out of our pipeline. So that's very exciting to see after all the hard work we've been putting in. So the Tree of Life genome engine looks something like this. We start off with sample acquisition in the top, work all the way through our pipeline until we end with the publication of genome notes at the end here. And the core lab sits within this blue box here, where our main focus is extracting DNA and high quality DNA and RNA from these organisms, and then getting them ready for PacBio sequencing. So delving a little bit deeper into what we do in the core lab. So when we receive a sample, we have a triage system where we weigh, prepare the tissue, for various downstream processes. So part of that tissue is sent for high-C cross-linking and sequencing with our colleagues in the scientific operations here at Sanger. And then we also prepare tissue for both high molecular weight DNA extraction as well as RNA extraction. And then those extractions from the DNA that pass get fragmented and prepared for PacBio high-phase sequencing. We have two methodologies of PacBio sequencing that we do here at Sanger. One is our low input or our standard input protocol, where we need about 500 nanograms of DNA. And then we have our ultra low input protocol, where we need roughly 20 nanograms of DNA. So there's two options that we can follow for sequencing. And in terms of high molecular weight DNA, this is something that we're looking for. It's a nice strong peak of high molecular weight DNA of sufficient yield that we could put forward to our DNA fragmentation and clean up before submitting these samples for PacBio sequencing. So when it comes to working with sort of very diverse taxonomic groups, I mean, each group presents various challenges that need to be met in order to obtain good sequence data. And one of these main challenges that we face in the core lab is obtaining sufficient as well as good quality DNA. And so this can be achieved by pursuing good sample preparation techniques. And an example of this that I presented in the past is how we prepare sponge tissue by applying what's known as the squeeze method. And this has seen a great improvement in the DNA quality that we're able to extract from sponge tissue. And it's also seen an improvement in high-C cross-linking and generating much better high-C data for the sponges. We also need to optimize extraction protocols for various groups. And so along these lines, today I'll be talking about R&D work that we've been doing in the core lab over the past year. One looking at DNA extraction optimization in the jellyfish, and the other is disrupting corals so that we can work with these for both DNA and high-C prep. So starting with the jellyfish, and I've included the comb jellies here because we have applied the same protocol to these. Our standard method of extraction in the core lab is to extract DNA using a QIAGEN MagaTrack kit. However, we found that when we have used this kit, we've found that we've been getting very low yielding, poor quality DNA. And the protocols that we've been using requires an input of 25 milligrams of sample input, followed by a 2-hour lysis at 25 degrees Celsius. This lysate is incorporated with magnetic beads, which bind to the high molecular DNA, and then we're able to elute that after a couple of wash steps. However, for the jellyfish, we were getting very low yields following this protocol. You can see here on the right that we have quality that is okay for proceeding to PacBio sequencing. However, the yields have been very low. So here we have 5 nanograms and 23 nanograms of DNA, which for us is not good enough for PacBio low input or even the ULI procedure. So following this, we decided to increase the sample input to 100 milligrams, and then we tried both our automatic and our manual extraction approaches to see if we could get better quality as well as high yielding DNA. So you can see in these profiles at the bottom here that our yields we were able to get high yields after increasing sample input, and that the profiles remained relatively stable compared to the initial extraction protocol. So generally, we would be able to use this DNA for our ultra-low input PacBio sequencing, which is a good thing. However, we didn't see universal success for our jellyfish. So here we have 4 other species, and you can see here we have cases where the yield is either very low, or in cases where the yield is slightly higher, the DNA profile was very poor. So we won't be able to get suitable DNA of good quality to submit for sequencing. So following on from the MagaTrack kit, we decided to try the EZNA mollusk DNA kit from Omega Biotech. And this kit uses spin column extraction methodology for mainly mollusks, but for other invertebrate tissues as well. And it relies on a CTAB-based lysis buffer followed by chloroform to remove polysaccharides. Then we also have our lysis steps at 60 and 70 degrees at various stages during this extraction protocol. So because the desired DNA yields were not met during our MagaTrack extractions, we decided to increase our input mass from 100 milligrams to around 200 milligrams. But as you can see, even though we were able to get really good yields from these extracts, so now over 2,000 nanograms, our DNA profiles were very poor. So this could be due to two reasons for this. The first obvious one is that we've been using spin columns, so this would probably likely fragment the DNA, and also our lysis temperature is at 60 and 70 degrees, so also another overlysing of our DNA, resulting in shorter fragment sizes, so definitely not suitable for any PacBio sequencing. And so the next approach that Amy decided to use was to add magnetic beads to this DNA protocol. So removing the spin columns altogether, and then also deciding to decrease the lysis temperature to 25 degrees, back to what we had during our MagaTrack extractions. And again, followed a chloroform isoamyl step to remove the mucopolysaccharides, and then adding magnetic beads to the lysate, followed by 2 ethanol washes, and then finally a DNA elution. And as you can see here, mixed results, I would say. So on the left here, we have an extraction that gave great amount of DNA, so 15,000 nanograms. However, the profile was pretty poor. However, on the right, we start to see our first sort of semblance of high molecular weight DNA in the jellyfish, and the yield was also very good. So not quite the heights of 15,000 nanograms, but around 1,500 nanograms. And from this, we were able to prepare 2 libraries, one for ultra-low input and one for low input. And from this DNA extraction, we were able to get 22 gigs of data on the Sequel2 and 90 gigs on the Revio. So for us, that was our first real success at jellyfish extractions, and enough evidence to try this on other jellyfish species that we have sitting in our freezers. So up to now, of the 31 species of jellyfish and comb jellies that we have as part of the ASG project, 22 have been submitted for low input and or ultra low input sequencing, with 19 having received sufficient PacBio coverage for the completion of the genomes. So moving on to the corals now, and this is work led by Ben. Corals are tricky in a different sense, in that we have to try access the coral polyps while preserving the genomic DNA integrity from inside the hard skeleton of the corals. And so this requires substantial force to pulverize the corals into a nice fine powder that we can use for DNA extractions. And so the types of disruption methods that we have in the core lab, we have the power masher, which is a sort of mechanized pestle and mortar. So we have a pestle that goes into a sample tube. We add the lysis buffer, and then we can disrupt the sample. Obviously, for the soft corals, this is potentially a way to go forward, but for the hard corals, we're going to get nowhere. So another method that we used in the past was the Covirus cryoprep, where we add samples into this cryotube. We place the tube into liquid nitrogen, pop the tube into this cryoprep, and then the sample is smashed by force onto an impact plate. However, the problem with corals is that they're so hard that, first of all, the bag often breaks, and then we lose sample into the machine. And then also the hardness of the corals has damaged the machine, preventing sort of further loss of other samples that have gone into the instrument. And so the next step we decided to try was the FastPrep 96, which uses beads into sample tubes and then shaken at a high velocity to disrupt samples into very fine powder. So we use the FastPrep 96 routinely to disrupt plant material, where we place 3 stainless steel beads into a FluidX tube, shake the sample at a very high velocity until it's a fine powder. But could a different bead, such as the zirconia bead here, work for the corals to disrupt it into a fine powder? So here at the bottom here, I have a picture of a coral pre-disruption. We then, on dry ice, add the bead and a coral to a polycarbonate tube, shake the sample in the FastPrep, and then out comes a lovely powdered coral sample that we can put forward to DNA extraction. So what are the DNA extraction outcomes that we see using bead-beaten material? So on the left here, I have an example of an extraction using our old cryoprep methodology. So another issue with the cryoprep is that often the sample doesn't disrupt into a nice fine powder, and there are often chunks of material left over that go into DNA extraction and then inhibits efficient lysing. So here's a good example of that. We only have 190 nanograms of DNA, and it's all very highly fragmented DNA and not suitable for PacBio sequencing. However, on the right, we have a really nice profile of high molecular weight DNA, and also a very, very good yield. So 6,500 nanograms of DNA. Again, I mean, this is not a...
Graham, I'm sorry to interrupt, but you're a little bit over time already. So
okay.
If you could.
Okay. I will finish. Thank you. So yeah, here's an extraction where we weren't able to get really good high molecular weight DNA of sufficient yield. Suitable for high C. We found that compared to cryoprep material, we were able to get much better high C signal from coral samples that were bead-beaten. So here we have a run of high C sequencing that's much closer to the example of good high C data that we would that allows for good scaffolding of a genome. So up to now, we have 41 coral and genomes are public or in curation. So in conclusion, over the past year, we've been working on optimizing sample preparation DNA extractions for tricky ASG samples. We now have a working extraction for jellyfish, and a more efficient sample disruption of coral tissues has seen improvement in DNA extraction and high C sequencing results for corals. Thank you very much to everyone who's helped in the core lab team, and especially to the Gordon and Betty Moore Foundation for their support of the ASG project. Thank you, and sorry for running over time.
Yes, no worries, Graham. Thank you very much for that overview. Is there a quick question from the audience before we move on? I mean, I can just jump in and say, I mean, it's just an amazing amount of technical, detective work, basically, that you folks have done. I'm guessing the channels for making sure this information gets into the public domain is through the genome notes, so there'd be sort of any specific technical details associated with DNA extraction would go in there. Is that right?
Yeah, we also have these protocols are, if not on protocols.io, we have a protocols.io page that everything that we produce will be put on there as well. So I think the jellyfish one is in prep, but the coral bead beating, I think, is on there already.
Okay. All right, thanks so much. Graham, back to you, Uda.
Yeah, thank you very much, Graham. Very exciting stuff here. And we are now moving on to Michael Dawson from Merced, and he will talk about symbiosis in 3D. So Mike, please share your screen. Thank you. You're muted, Mike. Please unmute yourself. That is helpful. Thanks.
Okay, how's that? Sorry about that. All right. So my goal is to give a really quick introduction to our project with Sanger Institute, the ASG project. It's called Symbiosis in 3D, and it's trying to understand the diversity and dynamics of pelagic symbiosis from a genomic perspective. I've got myself here as a talk, but a big team involved in this, so it's et al. All right. So the brief outline for the talk is a really quick introduction to the team, the project scope, the taxa and some questions, the project status as it currently stands, and then I'll just have one summary slide. And the idea here is really to give an overview of the kinds of questions that we're going to be able to answer through the ASG project, right, which were previously unattainable to us or inaccessible to us. So as mentioned, a big team. So we had 14 people in our hub. These are distributed on all of the inhabited continents of the world. The co-PIs were Anne Thompson and Kelly Sutherland and Aki Odira, and then essentially 10 other people distributed around the world. And we collected samples from all these places using a variety of techniques, such as blue water diving from aquaria and through submersibles. The project scope is pelagic symbiosis, as I mentioned, and the reason for this is, there are a couple of reasons for this, right? So the first is that aquatic symbiosis are poorly known, right? So this was part of the reason for the ASG project in the first place. The second is that of the aquatic symbioses that are studied, many of them are primarily benthic, right? So bottom-dwelling sessile. Yet the metazoan phylogeny reveals many evolutionary transitions from benthic to pelagic forms. So these are the dark branches here or underlined taxa. And that this transition is oftentimes, but not always, associated with becoming gelatinous, i.e. soft, transparent, having a low carbon to volume ratio. And that a subset of these taxa also have symbiotic and non-symbiotic lineages. So we're interested in trying to understand how these factors relate to each other. The symbioses that we chose for our project were bioluminescence and photosymbiosis, but of course we're interested in the cobionts too that would come out of this. So to think about the taxa and the questions. So the first example that I'll give is the pyrosomes and bioluminescence. So pyrosomes are pelagic tunicates. This is an example of pyrosoma. They're globally abundant. When they bloom in large abundances, then they can alter the nature of the ecosystems in which they occur. The bioluminescent mechanism has been debated. There's some evidence that suggests it may be endogenous. There's some evidence that suggests it might be microbial symbionts. And so this is one of the questions that Kelly and Anne in particular are trying to address. In all of these cases, we're not only collecting the target organisms, such as pyrosomes, which sometimes can be small, sometimes very large, but we're also going to contrast them with aposymbiotic forms and non-symbiotic forms to try to identify, the cause and mechanisms, the characteristics of the symbionts and symbioses. The second set of taxa questions concerns aceals. Here are the examples of what we've collected. So this is a genus called Haplodiscus. It's a relatively large aceal. It can be seen here on corals in two different environments. The small brown dots in here are aceal called Amphiscolops. It's really neat organisms, just a couple of millimeters long, but it swims in pelagic environments. It can capture and eat copepods, so it's quite an amazing organism. And so we're interested in this benthic pelagic comparison here. The third set of taxa are the cnidarians, right? So we have a couple of anthozoans in here, anemones. So this is Anthopleurus anthogrammica. This is of particular interest to us as a benthic comparator, but also because it has not only a symbiosis with Zooxanthellae, but also symbiosis with Zoochlorellae. And these may occur independently, or they may occur together. And of course, there are many other anthozoans that we can consider the very well-known corals in terms of symbioses. And so they'll provide additional benthic contrasts, as will ahermatypic and non-symbiotic forms. So the main taxa that we're kind of interested in, or form the largest number of species in our hub, are the medusozoans. And so the novelty in the Medusozoans is this part of the life history here, right? So in common with anthozoans, then there are benthic portions to the life history, and these may reproduce asexually, but if they reproduce via Medusae, then we get this novel pelagic form that has to deal with entirely different environments and entirely different body forms, right? So we're interested in the genomic underpinnings of this. A key characteristic of all Medusozoans, as far as we know, is that when they reproduce, whether this is via Medusae sexually or via polyps, coming back to polyps, that the symbionts separate. So there are no symbionts passed in the gametes, right? So they have to re-inoculate again when you get your new benthic polyp form. There is a lot of gain and loss in this clade. So sometimes there are Medusozoans that have the kind of like pelagic Medusa stage. Sometimes there are Medusozoans that don't, right? So they're both pelagic benthic transitions that we're interested in terms of life history. And these pelagic benthic transitions in life history are also reflected in the evolutionary transitions. So we see transitions from benthic pelagic forms at the base of the Cnidarian Medusozoan tree here, but there are also reversions from pelagic to benthic. There are at least 7 independent origins of photosymbioses in this group. So in cubozoans, in hydrozoans, there are several, and in cyphozoans, there are at least 2. And oftentimes these will be single lineages of hosts and single lineages of symbiont, but there are also cases where hosts will form symbioses with many different, with single lineages, and that there are a variety of genera of symbionts that also form associations with multiple hosts. So lots of interesting dynamic stuff going on here that we're interested to try to uncover. Here are some of the examples of taxa that we've collected for the ASG project. So this is a hydrozoan. These are fire corals, and they're one of the forms that has primarily a large benthic life stage. These also produce Medusae, but they're very small, and the major form that you'll see is this. And you can see that they come in different phenotypes, in different environments, and also across species. These are other capitate hydrozoans, but these are actually, turns out, colonial polyps colonial polyps that are pelagic, not benthic. So by the Winsela and the blue button, again, these have symbioses they're interested in, but again, variations on this general life cycle that I've shown to you. In the cubozoa, our second class, there is only one known zooxanthellates Medusa form. This is Alatina. And so we'll be comparing that with non-photosymbiotic forms such as Chironex. And then in cyphozoans, the main class that we're working with, there are 3 orders: the Symbiostomes, in which we know of no photosymbioses, the Rhizostomes, in which we have the main diversification of photosymbiotic forms, and then the Coronates. And the Coronates, kind of interesting, is primarily a deep water, large-bodied order of jellyfish, but the symbiotic forms are actually shallow water tropical forms in these here, and they have quite prominent benthic forms as well as really interesting pelagic forms. The rest of the Medusae on this image are Rhizostomes, and they include the upside-down jellyfish, which is this example of a reversion to a benthic Medusa form. Cotyloryza is interesting because it loses its photosymbioses when the organisms mature. And then Phylloryza and Mystigius come in a variety of forms, and we've seen both aposymbiotic and symbiotic forms of both of these species. And there's also patterns of rapid evolution in Mystigius, which are interesting. Okay, so there's a variety of differences between species, life history, habitats that may be related to symbioses. There are also questions about whether symbiotic forms that are marked here cause mass occurrences. You may have heard about jellyfish blooms. So we're interested in that interaction. We're interested in trying to use the genomes to better resolve these portions of the tree that are important in understanding the photosymbioses, and also in trying to understand the gross morphology in the internal anatomy of the jellyfishes that may be related to the functioning of the symbioses. So lots of work here. And then we're also interested in some like population level questions. So this is Mystigius that I just showed you a couple of slides ago. And there has been this really interesting occurrence where there have been both symbiotic forms and aposymbiotic forms occurring in the same place at the same time. And so these are marine lakes in Palau. This is the example. And so we were interested in whether the presence of the symbiont affected behavior. And the answer is yes, right? So golden jellyfish with the symbiont here are showing their distribution in the morning, their distribution in the afternoon, and you can see that changes. And that's because the animals swim directionally with the symbiont, so eastward in the morning, westward in the afternoon. But if you look at the non-symbiotic forms here, they show no directional swimming. So it seems from this presence of the symbionts is enough to change the behavior of the medusa, which is really fascinating. So we're interested in that. Okay, so where's the project's status? So as Gray mentioned, working with jellyfishes is not an easy job, but the Sanger project has done a really fantastic job. So we've submitted 39 systems. We collected another dozen more, but they were held up by export regulations. 27 are in data production. 11 genomes have been submitted to ENA and published. These include an A seal and an MNE and a suite of cipher zones. 6 manuscripts for the genome notes are in prep for this. In terms of the completeness of the genomes that are available, so the Sanger ones are shaded in gray here, others that are being produced by other projects are in white. The Sanger ones are slightly better than the breasts, so they're more consistent and more complete than other genomes that are available for jellyfishes. But it's really in terms of the contiguity where they are most impressive. So really strong N50s for the scaffolds, better than essentially anything else that's around. And this is reflected then in the small number of scaffolds that are present in the final assemblies. As few as 19 in Pseudorhiza, up to 1,000 in Cassiopeia, which is the worst of the set. But even this is twofold better than Cassiopeia produced by other projects, right? These are a lot. So really great high quality genomes being produced that we'll be able to work with. If you order these by genus, though, what you'll see is that when there are several estimates of genome size for generics, there's actually great variety in the assembly size, right? And this is more than we might expect within genera. So it seems like there's some work to be done here. If we try to compare the assembly sizes across all of these with prior estimates of genome size from, say, C values, there's maybe a slight positive correlation, but it's not strong. Maybe reassuring for our work with ASG is that the estimates from ASG come out as being kind of like in the middle of the pack, right? So they're not out in the extremes. It offers some kind of perhaps a sense of reliability. But it'll be interesting to see how this develops as we get more genomes from Sanger, including additional congenerics. And this kind of ambiguity with prior results also spans to our estimates of chromosome size. So in all of the ASG assemblies so far, there are 10 putative chromosome scale scaffolds. This is confirmed by the high-C analyses, whereas prior analyses of, say, karyotypes go from a diploid number of anything from 18 up to 42. So really highly variable there. So as we move ahead, we really look forward to seeing more of these 39 systems come online. It's going to be a fantastic resource. We've sampled about 5% of cipher zone species as part of this. This includes a mix of benthic, pelagic, symbiotic, and non-symbiotic forms. In addition to the reference genomes, we have geographical population sampling for about half of these species. And we have 11 host genomes available. They include the highest quality Medusa zone genomes available, 28 in the pipeline. We're starting to begin comparative analyses of these now. We're interested to diversify species, refine these estimates of, say, genome sizes, karyotypes, number of scaffolds as we move forward, and also start a suite of experiments and field observations to try to put the reference genomes to greatest use in understanding the ecology and life history of these and evolution of these organisms. So acknowledgements, this wouldn't have been possible without ASG. What an amazing group of people to work with, especially thanks to Mark Blacks and Victoria McKenna and How You. To folk in the lab and our support in the field, Coral Reef Research Foundation, and the population level sampling was supported by a NET and SF award and Division of Environmental Biology. If folk are interested to work on these, drop me a line. We would love to chat with you. Okay, thank you.
Okay, thank you so much, Mike. Amazing stuff. Question? Maybe I'll just encourage people to put their questions in the chat, and also people can if the speakers can have a look and potentially answer those questions. That would be great. Did someone have a question?
I have seen no question, but keep in mind the chat function is up and running, and we will therefore move on. Thanking Mike first and foremost for his really fantastic, inspiring presentation. And now we have Carlos Prada about speciation across depth gradient on, so we stay with the reef, coral reef environment. So Carlos, your slides
Can you hear me?
Yes, very
Can
well.
you hear me?
Thank you. Mm-hmm.
And are you seeing the slides too? Cool. Okay, so my talk is a little bit different because I will present actually the work that we have done, and I have a little less to say about the actual methods and the way the genomes were created, but I'm happy to answer questions if you have them. So my work has been mostly on understanding biodiversity, mostly on Caribbean reefs. And when you go to that typical Caribbean reef, this is what you see. You see a lot of diversity there. And sort of the diversity usually changes as you go from very shallow areas in that reef into deeper environments. And that has been kind of well documented, at least for coral reef ecologists. And so you have very different sort of shapes and forms in shallow waters. You have all these branching corals in very shallow waters. And as you go deeper into the ocean, you have very different forms. And actually, what is you are very, very deep into the ocean, then you start seeing all these platy-like shapes. And so in a very short stretch of water or space, you have a dramatic change in forms. And basically, or at least what we think, is that light has a lot to do with that major changes in biodiversity in such a small space. You go from very shallow areas in which you have a lot of light into deeper areas where you have something like 5 to 10% of the light that you have in shallow waters. And corals live in symbiosis, as you're probably very familiar here in this event. That they live in symbiosis with the dinoflagellate. So the coral itself is actually a very good cone that collects light for the symbiont, and the symbiont provides a lot for the coral. And so that association really needs to work very well, and it needs to be adjusted to the different light levels in which both the coral and the symbiont live. And so they are kind of in a fight in two areas. They need light so that they don't become limited by light, so their photosynthesis is performed. But if there is too much light, they could be light stress, or the photosystems could damage. And so they need a happy medium in the middle in which they have enough light to produce energy, but not too much to be bad for corals. And so that's what we have been working with shallow water corals on Caribbean, especially in the last few years with this coral, Orbicella fabulata, which is probably the most common coral now on Caribbean reefs. And if you look at this coral and you go across depths, these are about 100 genomes if you collect them from shallow areas to deep areas. So you swim from 5 meters to about 20 meters, and you start collecting all these corals. And this is what you see genomically. So you have one basic type in shallow waters, and you have a very different genotype on the deep corals. Also notice that in the middle layers, they don't really hybridize, or at least we don't see these mixed individuals that we see in regular hybrid zones, for instance, like bear hybrid zones. And they seem to be two lineages that are very recent. So these are population reconstructions, and we've done a handful of these for different corals, and we never go back more than 500,000 years. So these are recent events that have been produced in these very short spatial scales. Morphologically, they are also very different. So if you go from shallow to deep, again, you do the same swimming. What we're seeing consistently across species, both of hard corals, but also soft corals, is that you have an increases in columella or calyx size. And you also get a major decrease in the amount of those calyces. And so there is a major rearrangement of the calyx size and also the number. Our hypothesis is that in shallow waters, they become narrower and deeper, and that provides some sort of shading for to avoid light limitation. And in deeper areas, they are broader and shallower to increase exposure to light. Also, having less calyces, less density of calyces, avoid the self-shading of the calyces when you are deep. And we have recently, we kind of summarized of this data, and this is a process that is very common on reefs. It occurs in at least 14 different families of corals, both soft corals and hard corals, occurs across all of the major ocean basins. And so this is very common. And so the major question that we had when we started this project was, so what is producing all these different kind of species repeatedly across ocean basins and also across the Cnidarian tree of life? And obviously the closest area in which we could kind of start making progress was to actually look at light effects on behavior. And we also know from previous work by Nancy Knowlton and Don Levington that spawning, and previous other people, that spawning is pretty tied to light. And so if you have corals in different depths, they may spawn at different times. Orbicella or annularis, which is a close relative of the Orbicella fabulata, usually spawns a lot later than Orbicella franci, about 2 hours later. And that the reason for that seems to be related to the perception of light. Orbicella franci, which is deeper, is looking or is perceiving less light, and is perceiving sunset and moonrise a lot faster than Orbicella annularis. And so it seems to be that the segregation across depth could be related to this spawning behavior. And so what we did was to try to look at signals across the genome that may be related to this adaptation across depths or segregation across depths, and that may be related to spawning behavior. And one aspect that you see is that in an earlier figure that I show you, I show that there are clearly two different genetic backgrounds, a blue and a red background, and they look very clear when you do a mixture plot. But the fact is that for most of the genome, they are pretty similar. And you can see this in this figure. Most of the stuff is shared across the two different lineages that are segregated, or species that are segregated by depth. And only in a few locations, they are very, very different. And the locations that seem to be kind of segregated between them are G protein coupled receptors. And most of you or some of you may be familiar with this, but these G protein coupled receptors include rhodopsin-like genes and opsins in general. And there has been already a lot of work that has been done on this subject. And here is a model from previous work that shows all these protein coupled receptors. And in red, we highlight the ones that we have found segregated between depth segregated lineages. And basically our working hypothesis is that rhodopsin-like genes are capturing changes in light that is sending signals to the glutamate pathway, and that is opening channels that connect intracellularly to extracellularly, and that's allowing increase in calcium ions, and that calcium ions make a cascade into the camodulin pathway, and that's what's triggering apoptosis and the relaxation of muscles, and that's what we think is actually sparking the spawning behavior. And obviously, this is more like kind of correlational. So we are hoping to do more work related to actual expression of the genes. Nonetheless, we already have gene expression data that suggests that these genes are actually expressed pre and post spawning in the same coral species. And what we also seen is that they generate very sharp declines, and the selection in those genes seem to be pretty high. So if you take an heterozygote disadvantage model, is about 8% per generation, which is pretty high. And not only that, but they seem to be also under positive selection. So we have looked at a few of these genes, and they seem to be above, non-synonymous substitution seem to be higher than synonymous ones. And so it looks like opsin genes are actually related to those depth segregating corals that are kind of being produced in many different parts of the world and across the Cnidarian tree of life. And so with the new genomes, this is one of our hopes to see what is the replication of all these patterns in the different coral systems. And I think with that, but this project has been done with, the light aspects with Roberto Iglesias, and Don and Nancy have been involved in the work, in the genomics work, and Monica with the genomics work across depths, and the funders. And I take any questions if you have.
Thank you so much, Carlos. Fascinating. The questions are flying in the chat, but we do have a bit of time for a question. I guess if I could ask just this question of the timing of spawning and so forth as it relates to light and so forth. I just don't have a good sense for how all this is known. Is this based on detailed observation in the wild, or is it something that can be actually controlled experimentally in a lab setting?
So there is quite a lot of background in the field. So people from as long as they could dive, probably they have observed the correlation, or at least the correlation between changes in light and spawning behavior. And so it's well known that after sunset, a few hours, depending on the species, you spawn the gametes. But at least from 2 decades ago, or probably more, Don and Nancy have done pretty good detailed work on spawning behavior across depths, and they have already kind of established this system in which there is a difference between spawning timing depending on where you are in the water column. And so that's known. Experimentally, it's a little bit more difficult, but people have, shade corals and unshade corals, bring corals to labs and put shade and not shade.
Apologies for interrupting, Carlos. I'm keen on keeping the time window, and maybe this discussion could be put into the chat, because we have one more speaker, and it's Lucia Pita coming up. But I still wish you all speakers to hear the applause of 120 participants. Yeah, chat. So thank you very much. But we are moving to move on. I'm sorry, but I'm also looking forward to our last speaker, and that is Lucia Pita Galan from the University of Vigo, and she will have her talk on knot-like receptors in Mediterranean sponges. And I think it follows very nicely to what you have presented, Carlos. So Lucia, the stage is yours.
Thank you. Can you hear me properly, and can you see my slides, I hope? Yeah, thanks for the opportunity to share our ongoing efforts to dive into the diversity of this family of immune receptors, the knot-like, in Mediterranean sponges, thanks to the new data that has been put together by the Aquatic Symbiosis Genomics project. So we work on sponges that belong to the phylum Porifera. And
Lucia, do you want to put it in full screen mode? Your presentation is not in full screen mode. I think it will be helpful.
Okay, that's is now in full mode?
Yes, I
Okay.
hope, is it? There's a black box on the right. Yes, now it's in, yeah.
It is?
Yes.
I hope this
Thank
is
you,
working.
thank you.
Yeah, thanks, Ute. Thanks for letting me know. So we work with sponges that belong to the phylum Porifera, and is one of the earliest diverging animal groups. So whatever they found, every feature we found that is shared across this group and other animals may detect a characteristic of the last common ancestor. And they are also key members of the ecosystems where they are filtering water through their bodies, as can be visualized here with a dye. And they influence the cycling of nutrients in the system thanks to the metabolism of the sponge and its microbiome that is actually highly diverse. And in contrast to other animal groups, these microbes can be present in close contact to the animal cell. So they are a very interesting group to investigate the evolution of animal microbe symbiosis, and I'm particularly interested in the immune system that is emerging as the mediator between the microbe and the animal. And in particular, immune receptors like the NLRs participate in microbial recognition, not only in the context of pathogenicity, but also for successful and beneficial symbiosis. And these receptors will detect a signal and mount a specific response that will trigger either the elimination of a pathogen or tolerating a symbiont. But for invertebrates, we still really don't know very well the molecular mechanism behind. The NLRs are a receptor family conserved across divergent animal lineages from vertebrates to invertebrates, and with a function in defense and symbiosis that has been proved in experimental models. And they can be characterized in the different genomes by the presence of a conserved nucleotide oligomerizing domain, the NAC domain, that comes together with leucine-rich repeat motifs. The number of these motifs may be very variable. And this is what we call a bona fide NLR. But in animals, they commonly appear as a tripartite architecture with an N-terminal domain that is an effector domain, could be a death or a card or a period. And these leucine-rich repeats usually sense the signal, the microbe, and they can also auto-regulate the receptor, whereas the N-terminal domain will be the effector domain that then activates the downstream signaling pathway. And what we know in sponges is that this family of NLRs is highly expanded, at least in Amphimedon queenslandica, which is the species for which the first sponge genome was ever published back in 2004. But this has been the single sponge genome we had, and it was a draft genome. For about 15 years, we don't have any other reference. And despite the genome being rather small, there is a quite striking expansion of NLR. This is common to other invertebrates like sea urchins, for example. And this is the type of architectures that can be recovered, as well as some variations of this structure. So the question is whether this NLR expansion is common to all the sponges. What happens with these other novel architectures? They may play also a role in NLR signaling. And the question I'm more interested in is why this diversity could be a mechanism for a specific recognition. In 2022, Posadas et al. Took advantage of the new available transcriptomic data from different experiments to look a bit into the phylogeny of these NLRs, and they found some clustering based on the sponge class, although and it seems to be an expansion in those other sponges, except for Homoscleromorpha. They also look at patterns between what we call high microbial abundance and low microbial abundance sponges. So some sponges, they host a high density of microbes, whereas in other sponges, the microbiome is also highly diverse, but the abundances are much lower and they are more scattered across the sponge internal matrix. And there was no, it seems that there are more NLRs in LMA than HMA hosts. Regarding the domain architectures, they also found that those NLRs that were lacking the N-terminal domain were in generally more abundant, and there was no clear clustering based on N-terminal domain. But of course, this is based on transcriptomic data, so we are not sure if this is incomplete or also some different genes could be just fragmented genes. In terms of function, we don't have a sponge lab model, but we did experiments in aquaria with this species, this Idea vara, taking advantage, or we use RNA-seq, de novo transcriptome assembly, and differential gene expression analysis, and we found NLRs gene expression activated in response to microbial signal and different bacteria. So that's why we got interested in dig a bit more in this receptor group, because they may be actually involved in a specificity. And we started with getting some funding from the German Science Foundation when I was still in Germany, in a project we call ImmuBase. And among other things, we were able to generate a chromosome-level genome for this species, for this Idea vara. And this was mainly the work of the postdoc Pacifici Kosovel in collaboration with Monse Torres from the Genomic Center in Kiel. So we generated the DNA extractions, we do the sequencing, the high-seq we sent to a company, and we annotate the genome based on an in-house pipeline and supported the annotation with the RNA-seq data we generated years earlier. Well, we took time to go, let's say, in the detail of characterizing these NLRs. But what happens then, it was Aquatic Symbiosis Genomic Initiative. So now we went from a one sponge draft genome to be drowning on sponge data. So this is great. We wanted to test a pipeline to actually make use of this data to understand the NLR diversity on sponges. And we decided to start with some of the species that we actually submitted from the Northwestern Mediterranean Sea, and it was in collaboration with our local collaborator, Manuel Maldonado. We submitted around 12 species from this same location, actually collected at the same location. And among the first genomes from the Sponge Hub, and I would say, I think for the whole ASG, were these 5 species. There are more now already. Every month there are some updates, but we started working with this. I note that there is also this Idea vara. We decided to send samples there because we wanted to compare protocols, and also because we were interested in getting the metagenomes, right? And these different species cover 2 classes, Demospongia and Homoscleromorpha, and different host times in terms of HMA and LMA sponges. But note that each species belongs to a different order, so it's a bit like comparing a rat to a bat to a platypus, okay? So this is the taxonomic breadth we are working with. And we included also some 2 published genomes, one the one from Amphimedon queenslandica that has been the reference for all these years, and the one from Afrocalistes vastus, a glass sponge that was published last year, in this case at the chromosome level. This is how the assembly span for these genomes. It's all available by Sanger. This was really great. It's very easy to access this information. Just to notice that for Oscarella lobularis, we have the smaller genome size, and it's much smaller than what was predicted. So maybe there is something weird with the assembly or with this species, we don't know. And this is the protocol we use to look for the NLRs, okay? So we have these different species, but there are also a variety of annotation pipeline. And we look first for this NAC domain that is the most conserved part of the NLR. And for that, we built our own NAC profile based on some of the sponge sequences, but also other taxa, because we wanted to create a more diverse database than what is available in PFAM. And then we look for conserved domains based on sequence similarity with hammer search and smart search, but also based on structure similarity within interprostar. And we were looking first just to the bona fide NLR, so no other architectures that are these ones that you can see here. So this is what we have so far. In black, this bar, black bar denotes the number of NAC domain containing genes. So looking for those conserved, that conserved part of the receptor, and we have more than 100 in each of the species we explore. And for amphimedon, it's actually quite similar to what has been found by UN and collaborators in that paper that described amphimedon queenslandica NLR diversity. But then when we look into the bona fide NLR, so how many of these NAC domains are actually attached with the aleukin repeat motif, here the numbers start to change a lot, right? We have a less number of genes, and I find it quite interesting that we find more or a larger proportion in the IMU-based genome than in the ASG genome. And the explanation we have so far is that we have annotated these genes with RNA-seq data that comes from an experiment that was targeting immunity. So maybe this has helped to find the genes in the genome, but this needs to be tested. Also interesting, we didn't find any bona fide NLR in the homoscleromorpha sponge, and this was similar to what Posadas found in the transcriptome and data of other homoscleromorpha species. And overall, still we have an enrichment of bona fide NLRs. Here is where like the number of NLRs for human will be, but taking into account the human genome is much bigger than that of the sponges. So there is an expansion in all sponges, except for homoscleromorpha. And in general, in this tripartite architecture, the most common domain that we found was death. But we wonder if this is true, or maybe we are not good enough at capturing the CAR domain. And it seems to be a bit more NLRs in the LME than HMA sponges, but this needs to be tested. So in summary, we think that now we need to compare the number of recovered bona fide NLRs between the different gene models that are generated from the different annotations. So I saw now with Sanger there is always like news happening. So there are a new model that has been tested, ECALVA. So we have to download that and check our pipeline on that data to see if we have any difference. I think also very interesting is that we were more efficient at recover NLRs when we did this search based on structure similarity rather than sequence similarity. So this is something to take into account, and maybe where we should put our efforts in the future to try to avoid this manual curation. And we did detect transmembrane domains within the NLR. So NLRs were considered to be cytosolic because that's what they are in mammals. But in Iderian and in amphimedon already, they found transmembrane domain in bona fide NLRs, and we could confirm that. And as I mentioned, the NLR family is expanded in sponges, particularly in Demospongia. But this HMA, LMA enrichment, we need to test with more species. And with that, I would like to thank my collaborators, and in particularly today, the Aquatic Symbiosis Genome Hub, led by Ute and the Sanger team. It's been a pleasure so far working with you. Thanks a lot.
Well, thanks so much, Lucia. That was wonderful. I guess we're up against the end of time, Ute, but Lucia, there's a couple of questions already for you in the chat there. So if you could follow up there, and I suppose we can leave the channel open for a little while here so people can just follow up on Q&As as they wish. Does that sound about right, Ute?
Yeah, I think that's very fine. I think she deserves one question in spite of time. Sure. Maybe anybody has a question, now is the time to ask. And of course, Andia, please.
Hi, good morning, afternoon. Lucia, great presentation. I'm extremely curious about the annotation of sponge genomes, considering the replication of genes that we have. Like some of the expected genes, you think of an animal, oh yeah, this is the gene, and then you annotate. But in sponges, it seems to be that we have replications and replications and replications of some genes. So that makes very difficult the annotation. And I put actually a couple of questions there, but in considering that situation, when you are analyzing genome size or when you are determining specific genes that are of interest for, for immune system related, how do you manage that, because I think we have a big bias overall, and maybe it's across all invertebrates, who knows, but just want to see what is your point of view with that bias of trying to define.
Short question, please,
things.
Andia. I'm sorry to be brutal here, but the next session is ongoing. So a short question, short answer, please.
I think the short answer will be that we need to test the different annotations, and then we need to decide to what extent we want to go to manual curation. So maybe for specific gene families, we need to do that work.
Okay, and do your comparison with Ephemeron queenslandica are the original annotations of that species or new annotations of that species?
So this was with the original annotation, and we did recover less NLR than that UN and collaborators because they actually look for different gene models and they do a lot of manual search. But we wanted to test how good we could be with this more automatically search.
Yeah.
So this is not the end of the story, okay? This is actually the beginning.
Yeah, thank you so much. The chat will continue a little while, so please feel free to keep going. And again, a great thanks to all the speakers who prepared and to the 120 participants we had here. I think John and I are most delighted, and I hope that John and I could spark the excitement into the community as much as Zoom allows us. Hope to see you soon again and keep the inspiration. Keep it going. Bye-bye. Thank you,
Thanks,
Hayu, for
everyone.
hosting.
Thanks, Ralph.
Thanks to the Jam for organizing.